I like doing the first slot after dinner because you sat there with all that food in your stomach. Yeah, we've opened the windows, but you're probably not completely awake. What I want to point out is that if you're half asleep, these aren't dogs. These things with big fluffy ears are horses. I know far more about horses and equine genetics than I do about canine genetics. I make no apology for that. But what I can say is that my background is not in academia. First and foremost, my partner and I are horse breeders. I ignore the West Middlesex accent. I was actually brought up down in Devon. I learned to ride when I was about 9 years old. I used to point to point like Thomas did down there. And then as I moved on, now living in Cumbria on a small holding in Hertfordshire, and for many years we kept and bred Cleveland Bay horses. The other part of my background, the story I haven't told you so far, is that I'm a qualified teacher. I'm a teacher of biology, but of outdoor education as well. My love is teaching kids in that best classroom of all, outside of the bricks and mortar. But as a biologist, when Jill and I first got involved with doing Cleveland Bay horses, which we knew were rare, with a scientific thing, I started to ask the question as I'm shuffling around stud books on the table of the night. How do you breed these rare breed horses for the betterment of the breed and not just for what I want to do? And so I look around and I look and see what science there is round and about. And this is obviously in days prior to the internet. But when you do the research, you actually find out that the real research on what to do with rare breeds doesn't stem from canine biology, doesn't stem from equine biology or any aspect of livestock biology. It actually comes from the world of management of captive wildlife. You know, really, I think animals that we perceive as being really rare, like white rhinos, Siberian tigers, giant pandas. And the reason that the science comes from there is because of Walt Disney. Walt Disney made a huge amount of money out of making films about fluffy animals. And he pumped a lot of that back in to research about how to manage them in zoos, pumped it back in through the American Zoological Association. And they in turn funded research at the Smithsonian and at Lincoln Park Zoo. And they came up with the science behind how we manage captive populations of wildlife, and it's been taken on and adapted to manage livestock and to manage other domestic breeds. So that's my background. I'm also on the expert advisory panel for the Rare Breeds Survival Trust, which is an umbrella charity. It's an umbrella charity that looks after our rarest breeds of livestock, including equines, and it in turn has a responsibility to report the state of our livestock biodiversity to DEFRA, to our government. And that's handled through a body called FANGA, Farm Animal and Genetic Resources Committee, of which Philippe, who's sat at the back of the room, sits on that government committee. Government in turn has obligations. And we heard earlier on today about how well the UK is doing in terms of managing plant biodiversity. Government has targets to meet set by the Food and Agriculture Organization of the United Nations. API target number 13 is about maintaining biodiversity, and we are meeting, and more some, our target in relation to plant biodiversity, mainly because of the seed banks that we've got. We are nowhere near with our livestock biodiversity. Okay. So what I want to cover today is a real quick primer on conservation genetics. What's important, what isn't so important, what do we need to do with any of these rare breeds, be they in a zoo, be they livestock, or be they domestic pets. I want to talk about the problems that we have specifically once you set up a breed book, a stud book, and you say, right, this is our breed, we're going to draw a ring fence around it, and we're not going to have anything else coming in, because that in itself brings its own genetic problems. I'm then going to move on to look at strategies that can be used to help deal with those problems and to counteract those problems, and then to look at how we continue to monitor our breeds and manage them and secure them into the future, because that's what we all want to do, whether it's cats, dogs, mice, horses, or rare cattle. We just want to be able to preserve our breeds into the future. So, whether it's an air escape pony or a rare breed dog, you will have problems with a small closed population. And the biggest problem of all is that they will lose genetic diversity. That's inevitable, but they will lose genetic diversity faster the smaller the population is. And that's through a process called genetic drift. There's nothing you can do about it. It happens, but it just happens faster the smaller the population is. And I'm going to go into a bit more detail about what genetic drift is and its consequences in a moment. I'm sure because of the investment that Kennel Club has made over the years in focusing on inbreeding, you've all heard about inbreeding and about these what we call deleterious traits that build up. That's a process called inbreeding depression, and we'll examine that in a moment. And with inbreeding depression, we actually accumulate these damaging genetic traits, and the breed as a whole will lose fitness and ability to survive into the future. Okay. Whether it's human beings, dogs, or horses, we are made up of DNA. DNA, double helix. At the end of each bar of the ladder, if you like, there are two genes, two copies, two alleles. And if we stripped all of those off, if we imagine it was a big pile of Smarties, if you tipped a Smarties tube out on the table, you would have a lot of different random colours, different coloured genes. But you would have a lot more of some colours and far fewer of the others. And in this example, we've got a lot of red Smarties. They're the common ones in the tub. The green ones are, rarer, and the blue ones are rarer. So they're all got different frequencies. And there are consequences because some are common and some are rare. Let's look at a mating of a stud dog and a bitch, or of a dam and a sire in the equine world. Stereotyping that will go pig for a girl and blue for a boy, okay? So animals carry two copies of a gene at any position. So at any one position out of, in equine terms, 2.4 billion potential places on the DNA. That's how big the equine genome is. But we're just looking at one place on that rung, one rung on that DNA ladder. The dam is carrying, let's say, A and B, and the sire is carrying C and D. So we've got C and D, two copies, but they're two different copies being carried by the dam. Let's do two theoretical matings. So in the first mating, whoopee, we've got a filly foal. Everybody wants a filly foal. A has been passed into the filly from its dam, and D has been passed into the filly from the sire. A and D are in that next generation. Okay, another breeding season comes round, we mate the same two animals. In this case, what's happened, we've got a colt foal. It's a 50-50 chance unless you go downstairs and use sex semen. In this case, the mare has passed B on to the colt foal. The stallion has passed B on. Now, if you look what we got, in the first generation there, we had A, B, C, and D. In this generation, we've now got A, we've got B, C is completely absent, and we've got two copies of D. We've, in other words, increased the frequency of D, but we've lost C. We've still got the same number, we've still got 4 genes, but we've got different frequency, and we've lost one. That's basic genetic drift. Okay. So if we started in our petri dish or in our animals, let's suppose we start with 10 genes and we do a number of generations of random mating, and we sample it somewhere down the tree at halfway house, we've still got the same number of marbles in the pot, if you like, but we've only got 6 different colours. And if we carry on doing that random rating, several generations left, you can see what's happened is we've now got a dominance of red, a minority of blue, and all of the other colours have disappeared. And they've disappeared through that purely natural process of genetic drift. The smaller the population, the faster that will happen. And it happens generation on generation on generation, not year on year on year, generation on generation on generation. So in equines, the generation interval, certainly for the bigger horse breeds, is about 9 years. I'm kind of guessing, putting the thumb in the air, but I would say in canines it's probably 2-3 years, something like that. So it will happen faster in humans in calendar years. It will happen faster in canine breeds than it will in equine breeds. I was talking to somebody just now about the actual digging into the DNA as opposed to pedigrees, and with all our rare breed equines now, they're parentage tested. They're verified by DNA analysis, but they're not looking at the whole genomic profile. That would be extremely costly, and it would take a hell of a lot of time. It started in around about the late 80s, early 90s, where they could do blood tests, and then it's moved on to testing hair follicles, and they test what they call a panel of microsatellite markers. It's a tiny portion of the DNA, but the International Society for Animal Genetics has determined and identified 17 markers with which we can verify parentage in equines. So certainly for all the rare equine breeds, they're tested before they're registered and put in the stud book. That way they can say, going forwards, the stud book is as good as DNA, it's validated. It's a bit hard to interpret. You can see that's quite old. It's from an old dot matrix printer. So what I've done is I've just translated that into a clearer to read table. So we're looking at 16 markers. This is slightly earlier. Not all the markers have been tested, but we're looking at 16 here. The first one, VHL20, it's just the enumeration of one of the satellites. If you look, some of them are AHT5 and so forth. That was Animal Health Trust, yeah, who obviously did a lot of work with canines before it meant to be fun. But if we look first at the very top of the table, the sire, the 2 copies that he's carrying at VHL20 is I and N. So the sire's in the left-hand side of that table. If we jump across, sit in the middle for the moment, the dam, the 2 copies that she was carrying, are I and P. They've had a foal, we tested the foal. What's the foal carrying? So, the foal is M and P. Is that possible? Well, look, that M must have come from the sire because it's not present in the dam. That P must have come from the dam because it's not present in the sire. But yeah, that works. And if you carry on looking down that sheet, oh, the next one, that's interesting because both of the parent animals were KM. There was a bit of variation. They had 2 different copies. Yeah, but look what's happened here. The progeny's inherited M from the sire and M from the dam. It's said to be homozygous. If you've got different copies, you're heterozygous. If you've got identical copies, you're homozygous. And at that position, at HTG4, the progeny is homozygous. But if you carry on counting down and looking, you can actually see the parentage side of things works here. It's possible. So that animal's put in the stud book. We know what its parents were. This is, again, a real-life Cleveland Bay case. That's why I've blacked out the things. This is parentage not working. So the person's put in the registration. We've already got the dam and the sire on record. We've got a new hair sample for the foal that they've said is by this sire out of this dam. Look, let's see what happens. Back up at VHL20, the progeny is N and P. Well, is N possible? Yeah, it's possible. Is P possible? It's not there. Keep on working down, and actually there's quite a lot of things that don't work in that one. So that's a case of going back to the breeder and saying, right, what really happened? And actually, whilst you thought that stallion was covering that mare, that colt over there that's still entering has jumped the fence, and that's what's happened.
It was the donkey.
It was the donkey. So that's the principle of parentage testing. It isn't necessarily quite as applicable in the canine world. It would work, but that's against the panel of equine markers. What I want to show you now is what happens when you mate two animals that are very closely related and carry out a highly inbred mating. I've tabulated this. This is for real. Back in the mid-1990s, I can tell you now that both of the parent animals are dead and long gone. I've never identified who they are because you bet your bottom dollar, you know what breed societies are like at the start of an argument. But let's just have a look and see what the sire's carrying, what the dam's carrying, and more importantly, what's happened in the progeny. And the parentage works out here. These were the parent animals. So don't go working down the table, working that one out. What I want you to look at is the number of markers that are heterozygous and the ones that are homozygous in the parent animals to begin with. So if we look down the sire and do a countdown, what you actually find is the sire was homozygous for 6 out of that 16 markers. If you do the same for the dam, the dam was homozygous at 3 of those positions. Now count down the middle. Anyone got a stab on it quickly before I press the button?
8.
Any advance on 8? 10? Any advance on 10?
12.
It's 12. Okay, so in one generation, what has happened is we've lost a huge amount of diversity at those 16 markers. Well, hang on a minute, that's just 16 out of 2.4 billion markers within the whole equine genome. What the heck's going on in the markers that we haven't tested? So how much diversity have we lost as a result of that single mating? If we go back to the Smarties or the bag of marbles analogy, what did we start off with? We've got 2 animals, 16 markers, and they're carrying 2 copies of each marker. So we started off with 64 different marbles. And if you add up how many different coloured marbles the parent animals were carrying between them, they actually started off with 35 different colours. The sire brought 32 different colours. Brought 26 different colours to the party. The dam brought 25 different colours to the party. But we know that that homozygous progeny has lost a lot of diversity. In actual fact, yes, it's still got 32 markers there because we're testing 16 places. Yeah, but there are only 20 different colours. So in other words, if you carry out that mating, you were losing your marbles. The rarer colours have gone. Scale that up, that's one potential mating. That was a real mating, that really happened. We've now got highly inbred progeny. Diversify slightly. Is all inbreeding bad? No, it isn't. Inbreeding is how we set up our breeds. It was line breeding. Where does one stop and where does the other start? It's an interesting discussion. Let's look in a bit more detail at inbreeding. An individual is inbred if its mother and its father share a common ancestor. So you've got an inbreeding coefficient. Yeah, it's how related your mum was to your dad. What common ancestors did they share? Either recently or in the distance. You know, it might be right back at the time of the Battle of Hastings and the Northern Conquest that they shared some common ancestor. But that means that they've inherited identical genetics down both sides of the pedigree, and they're represented in you. It was 1922 when an American botanist called Sewell Wright actually came up with the concept of tracing those identical pathways in plant biology, and he came up with the idea of an inbreeding coefficient. So you can blame Sewell Wright for everything that follows and for all of the problems that we have. In genetic terms, let's do animal matings like we did before. Let's take 2 totally unrelated animals, so they do not share any common ancestors. And if we mate them, okay, so we're mating animals 1 and 2 to produce that filly foal, and then to produce the colt foal, those animals, although they've got identical parents, they're not inbred because the parents were not related. So that's a non-inbred generation. But then, if you take those 2 non-inbred offspring, so we're taking this filly foal here and this colt here, and mate them, Then we've got common ancestry, they share common parents, and then we're going to get inbred progeny. The table on the right-hand side, we're going to see it a little bit later on in larger format. That's from Sydney University, talking about the deleterious traits that accumulate within different types of livestock. But what does inbreeding do? Inbreeding actually reduces genetic diversity. We saw the loss of diversity that happened down that central column, that we only ended up with 20 different coloured marbles, whereas we'd started off with 35, we lost them in one generation. What does that do? It reduces genetic fitness. Now, do I necessarily mean the ability to run a mile? Well, actually, they've proven it in thoroughbreds, it does. Yeah, more inbred you are, the less chance there is of you appearing on a race course in the first place, and then your performance will not be as good as less inbred animals. It greatly increases the chance of you expressing what we call deleterious alleles. And we'll look at that in a moment. But certainly in the equine world, we know that there are at least 400 deleterious traits, things like hoof wall separation in Connemara ponies, lethal foal syndrome in Fells and Dales, where foals are born with an immunodeficiency syndrome that means they're unlikely to survive the first 24 hours, and if they're really lucky, they might make it to 2-3 days, but they're going to end up being put down because they just will not survive. And that's a deleterious trait. We know there are at least 258 and possibly as many as 46,000 in the equine genome. That's a huge number. So, yeah. There are documented, in the figures that I've got from 2018 prior to the pandemic, there are over 800 well documented and proven in dogs. The number will almost certainly be much greater than that, but that's the ones that are documented and in the database at Sydney University. And then the other thing that we need to think about is, yes, these animals might be inbred, but is the inbreeding from really, really recent in the pedigree? Was it full sibling mating, a cousin mating, or was it deep in the ancestry? Again, in human terms, was it common ancestors back at William the Conqueror? And from studying stud books, that really, really deep inbreeding can be almost impossible to detect. Tallis has already mentioned that my involvement was with Cleveland Bay horses and trying to deal with inbreeding at 22%. If you look at stud book registrations and then look at the pedigrees and work out inbreeding coefficients of what the animals were back in those days, if we look from 1900, the stud book itself goes back to the 1700s, just as long as the thoroughbred stud book does. But from 1900 onwards, you can see that inbreeding was increasing up to the time of the First World War. And then it suddenly decreases. Well, hang on a minute, that isn't biologically possible in the current population. Where did that come from? What actually happened was there's an amalgamation of two stud books. There was the Yorkshire Cultural Society stud book and the Cleveland Bay stud book. They decided that actually, you know what, they were one and the same thing. The numbers in the Yorkshire Cultural Society have gone right down the tubes. Let's just amalgamate it. But that's why there's a decrease. That's biologically not possible within a closed stud book. But it did happen. So we're now in the interwar years. And what happened with Cleveland Bays is they're numerically challenged. At that time, they're numerically challenged because so many went as war horses. They were the best of the best artillery horses. Shire horses are great. They can pull huge, heavy weights, but they can't do it fast. What do you want to do with artillery? You want to fire the gun, pack it up, shift before you get targeted yourself, and go and fire from somewhere else. And if you're doing that in the mud and the trenches of Flanders, there's only one horse that was really going to do that, and they were the best of the best. The problem was, very, very few of them came back. They didn't make it back to this country. So that's one of the reasons that Cleveland Bays became a rare breed. If you look at what happened, though, after the Great War, inbreeding, if we assume it was at zero then, it probably wasn't, but you can see that it's increasing in a pretty linear rate. But where does it get to? That's 0.1, that's 10%. Up here, we're at 20% and carrying on. So that by the turn of the millennium, we are in excess of 20% inbred. let's just put things in context. If you mate full siblings, brother and sister, that's 25% inbred. If you mate a mother and her own son, that's also 25% inbred. Father and his daughter is 25% inbred. We're getting up towards that. There's a really good reason as human beings we don't marry our cousins. Yeah, that's 12.5% inbred. A second cousin mating is 0.625. Okay. I don't know if any of you watched that film Deliverance, Julian Banjos. Yeah, great sequence. Yeah, the guy that's sitting there playing the banjo with the cauliflower ear. Say no more. Okay, that's where Cleveland Bay's were. If that continued, it's going to go way past 25% really quickly, and you can be 100% inbred. All right, yeah. Just because 25% is full brother, full sister, if they're sharing common ancestors as well, you can become more inbred than that. If we look at some other equine breeds, Exmoor pony, the green line is what we call average genetic relatedness. That's something to do. I'm going to talk about kinship in a moment, but in terms of inbreeding, the purple line is the one that's important. Again, you can see it's increasing in a big linear way. Then it approaches 15%, and the rate decreases rapidly. It's still accumulating, but you've got a plateau in there. That's quite interesting. If you look at Dale's ponies, again, that has increased dramatically, and then we've got some lumps and bumps. What that's about is actually fell ponies being brought into the breed. Yeah? So once you open the gate and let something in, it will have an effect. Is that the answer? No, it isn't. Just looking down some canine breeds, before I was asked to, when I was asked to do this talk, I just thought I'd do some comparisons. And if we're looking at kennel club data, I'm not putting those in any particular order or picking any particular breeds, but if you look at pedigree-based information, Airedale Terriers 17%, Nova Scotia, duck trolling dogs at 1.6, there's quite a range. You can also look at inbreeding from studying the DNA, molecular-based inbreeding. And that will often pick up a figure, that will truly tell you what alleles are being shared in common. And this is a study done very recently, 2021 study, looking at 227 breeds, and the average that they found there was 24%. So we're right back up there at full sibling mating level. That's the average across 227 breeds when you look at it at molecular level. So we're not looking at the reliability or the unreliability of pedigree data. This is genetic data. This is DNA telling us this. And you can see that even within that cohort of dogs, there's quite a range, so that we've got some that are still down at 0. We've got the majority around about 24% inbred, but we've then got some that stretch up through the few, nearly as high as 60% inbred. Again, just comparing dogs versus horses, and these are both molecular studies. The one at the top is from Peterson over in the States. She's obviously looked at far fewer breeds than the canine study underneath. This was 2013. And you can see that the lines have been drawn there. That second cousin mating is in green. The full cousin mating at 12.5% is in yellow. And the full sibling mating is up there in the red. You can see that that time they've identified that Clydesdales and Exmoors were up there close to that figure. Cleveland Bays don't feature in this table, I don't think. If you look at dogs, what we can see is that there are far more canine breeds up above that red line. And that study stems from data that was done just prior to the pandemic. That's 2018. We've seen this diagram in a whole variety of different forms today, people talking about this extinction vortex. If you've got a small population, yes, we know that inbreeding will accumulate. We also know we will lose diversity through random genetic drift. So as we carry on mating over a number of generations, we will lose genetic variability. And that will bring about a reduction in individual population, individual fitness, and in population adaptability, which means you've got a lower reproductive rate as one of the consequences. We've got higher mortality. So as we carry on round, we're going to end up with an even smaller population. And if you start with an even smaller population at the bottom of there, there's only one place this breed is going to go unless we do something to intervene, and that's down the plug hole. Let's look at deleterious traits and what does it mean. We saw a very similar diagram before when I explained in reading with the A, B, C and D at the top. We've still got the same dam, but we've got a different sire on the market here now. The sire is carrying C, but instead of carrying D, he's carrying X. And X is a genetic nasty. He's a carrier for one of these deleterious traits. Let's suppose it's a fell pony. He is a carrier for lethal foal syndrome. You can be a carrier, but as you and I look at it, standing there, you're looking at a perfectly healthy fell pony. You won't see anything wrong. It's only when two carriers are met together that there's a chance that we might physically see something. Let's look at the genetics of what happens. Right, so we're going to make this non-carrier with a carrier. X is the rare deleterious allele. What's passed on? The dam's passed A. The sire has passed X, so this filly foal is a carrier. Do the same mating again, just like we did before. This time, the dam has passed on B. Again, the sire has passed on X. They're both carriers, so both of the progeny in the second generation are carriers. Again, you won't see it if you line them up. They'll look like perfectly healthy foal ponies. It's only when you come to mate them, make two carriers, that there's a potential problem. So let's mate them. We know it's going to be inbred. It's going to be 25% inbred if the parents were unrelated. The dam in the second generation has passed X on into the third generation. This colt has passed B on into the next generation. We've got another carrier, B and X. Again, look at it, it will look fine. But what's happened in this next crossing is that this third generation animal has inherited two copies of X. This is the foal that isn't going to survive the first 24 hours. Okay? And you can extend that onto any of the Mendelian inherited recessive genes of deleterious traits. If you make two carriers, there is a chance of getting animals expressing the deleterious traits. So if we make two animals that are completely clear, the progeny can only be completely clear. You know, there's got to be carriers present. If we make one carrier with a clear animal, 50% will be clear and 50% will be carriers. Again, you will not see it because there are not two copies of that deleterious allele in the mating program. Right, now then, if we make a clear animal with one that's affected somehow, whatever it wasn't a lethal 24-hour lethal syndrome, these animals survive and it's gone on to mate in its own right, 100% of them will be carriers. None of the progeny will actually express the deleterious gene because there aren't two copies, but they will all carry it. Let's make a carrier and a clear 50-50, carrier and carrier. This is the situation we just had. We've got 50% will be, 25% will be clear, 25 will be affected, yeah, and 50% will become carriers. So we've now got a quarter of your foal crop, or a quarter of, there's a 25% chance that the animals that you breed will carry and express. They will have two copies, they will have that deleterious allele, and so on. Quite often thrown back at me when we say inbreeding is bad is what about Chillingham cattle? These are pretty much wild, feral herd of rare cattle that exist over at Chillingham in the northeast. They are highly inbred. These animals are nearly clones of themselves. You know, that inbred. Why do they survive quite healthily? Well, the reason they survive is because of a process called purging. You know that during COVID changed, right throughout the pandemic. We ended up with the Delta variant, the Omicron variant. Genes change. They're doing it all the time. We get mutations and they accumulate. At the same time, there's another process going on that's purging them, getting rid of mutations. And what's happening in chilling and cattle is because there is minimum human intervention, it's pretty much random mating. There's not a lot going on in terms of human intervention. Is the rate of accumulation of these deleterious alleles through inbreeding is being exceeded by the rate at which they're being purged. When I did my PhD, I was being supervised by Professor Stephen Hall, who's the expert, the world expert in chilling and cattle. And I said, why can't we do Cleveland eggs like this? Because we can't predict them. You cannot predict which populations will purge faster than their accumulate. It can happen. Not all inbreeding is bad, but for the vast majority of breeders it is, because this won't happen when there's big human intervention. Right, we've talked about inbreeding, about how related I am to myself through my mum and my dad. What I want to do is talk about kinship, because kinship is how I'm related to anybody else in the population. So it's how related I am to you through some distant ancestor and to you through some distant ancestor. And somewhere the chances are that there's a link. So it's the relationship between two individuals. Okay? Just like we can work out an inbreeding coefficient by looking down the pedigree and tracing the pathways and doing Sewell-White's calculations, we can work out the kinship coefficient as well. Full siblings might not be inbred, but they're 50% related. We've got common parents, yeah, mum and dad. So full siblings, even if they're not inbred, can be have a 50% kinship. We just look at that diagram again. Two unrelated parents, mated. F is the symbol for inbreeding, okay? F is still normal. These animals aren't inbred, but yet they're 50% related because they share the same parents. If we now make them, we already know that the inbreeding coefficient is going to be 25%, but they're actually 75% related. And you can see how that accumulates down the pedigree as we continue those crosses. Right, inbreeding is how related I am to myself. Kinship is how related I am to any one of you. Mean kinship is how related am I to everybody else in this room or to the rest of the world human population. The mean kinship. So it's work out all the individual kinships and then average it out. So it's the average kinship coefficients between an individual and everybody else in that living, that's important, the living population. So it's the relationship of me to everybody else, or of your dog to everybody else living within the studbook population. It's a good indicator of genetic diversity. If you look at the mean kinship of a breed, it gives you a good indication of where its genetic diversity, how much genetic diversity has it actually got. Individuals with a lot of living relatives are going to be high mean kinship, and that means that they're going to be carrying the animal, the genetics that are common in the population. An animal with a low mean kinship, they're the ones that are going to be carrying the rarer genetics. High mean kinship, do you remember the big pile of red Smarties? They're high mean kinship animals. And out on the shoulder, the blue Smarties, and I think was it the green Smarties at the other end, they're low mean kinship animals. If we look at a canine breed, and I picked this one at random, I have no idea whether it's relevant to you guys, but the Leon bird dog, if you look at what's happened to them since the 1900s, inbreeding's in green on this graph, you can see that it's increased in a pretty linear way, right the way through to around about the 1970s, and then it started to tail off. Interestingly, if you actually look at what's happened to kinship and to mean kinship of the breed, the whole population, that was decreasing here, but then look what's happened. It's actually ahead of inbreeding. Yeah, it's above it, it's above it, it's above it, and then it drops. And what happened, what we tend to find, is that inbreeding will follow mean kinship. If mean kinship starts to drop, or the rate of accumulation of inbreeding, mean kinship starts to drop, some years later, some generations later, inbreeding will also start to decrease. The rate of increase in inbreeding will reduce. So inbreeding follows kinship. Right. One more biological concept I want you to get the idea of, and that is a population size. How big is your population of your breed of dogs? How big is our Cleveland dog population? You know, when we look at idealized populations, they're huge. They're infinitely large. They do what chilling and cattle do, randomly mate. Every animal gets a chance to and does contribute equally to the next generation. Generations don't overlap. They're all nicely sorted. Yeah, they're all conceived on the same day. Yeah, and it all works out really neatly. Yeah, let's get real. In the real world, populations, whether it's livestock or domestic animals, domestic pets, they're restricted size because we can't put everything, right? Yeah, we've got unequal sex ratios. Certainly in horse breeding, maintain a load more dams than you do stallions. Okay? Unequal family sizes, and there's human selection. Yeah, we've all got our eye, with breeder's eye. You know, we know what we like, we know what the breed standard is, and we make selection. And of course, there's overlapping generations. It isn't tidy. So, as biologists, as geneticists, we have to come up with some sort of a measure of what is our effective population size. I was at Shire Horse Conference last weekend, and I was saying to them, right, what's the world Shire Horse population? And they came up with a figure of about 3,000. But I went, have you really got 3,000 horses? Because if you think of their genetics as Lego bricks, when we engage 4 or 5 3-year-old kids, we pull all those Lego bricks apart and stick them. No, we can line up those 3,000 horses over there and go, that's a big population. Take the Lego bricks and give the kids the job of sorting them out. How many unique animals could you build out of that pile of Lego bricks? They were really surprised when I said, you can probably only build 75. So you've got 3,000 sets of hooves on the ground, but you can only build 75 unique animals. And that's the sort of the analogy. And it's a measure of how much genetic drift is going on. Yeah, how fast genetic drift is reducing your diversity. So it is a really important parameter, whether it's canine breeding or equine breeding. It translates to the census of a real population that is losing genetic diversity at the rate of your population. And we can measure it in a number of ways. The gold standard way is to measure it in relation to the rate of accumulation of inbreeding. Remember I showed you the graph of Cleveland Bays with the red line? That was worrying because that really reduces effective population size. The other way that you can do it is by comparing the number of males and females that are breeding in any one generation. That's called the census effective population size. But the only real true way that isn't an adulteration of what Cyril Wright worked out in the first place is that you measure it from the rate of accumulation of inbreeding. So it's not the absolute level of inbreeding that is of particular importance. It's the where do you go from here? How fast does it continue accumulating from today? What's its history? How's it got there? And how fast is it accumulating in the future? It's great. I love the donkey. 5,500 rule. If you measure inbreeding from the rate, effective population size from the rate of increase in inbreeding, it needs to be greater than 50 for short-term survival. It's recognised now that if your effective population size is less than 50, it's very unlikely you've got a viable population. If it's less than 500, measured by census population size, the number of breeding males and breeding females, if it's less than 500, it's not likely to survive in the long term. Now, there is some argument about this, and I know there are some biologists in the room. There's an argument now that it should be 100, 1,000. But if we stick to the original, there's a reason for that, and that's because the United Nations still stick to 50. But at least 50 for the short-term survival of the breed, to avoid breeding depression, 500 for long-term survival. And that originates from back in the 1980s. And in order to be able to work out your effective population size, you need to understand your population, and stud books are really useful. Certainly in terms of breed societies in the equine world, they all maintain a stud book, they're in control of their own data, and we can crunch it. We can crunch those numbers through software like this population analysis report called PopRep, and it will tell us where's the inbreeding now, where was it historically, how's it accumulating, what's the effective population size. It also gives us straight figures about, you know, popular size, popular dams, and all sorts of demographics. But that's the key one, is we can say what the effective population size is. Is the breeding decline genetically, or is it stable, or is it improving? If we look at some canine breeds and where they are in terms of effective population size, this is a study of mean registrations from 1980 to 2014. This is UK data, okay, from Kennel Club data. It's a logarithmic scale, folks, don't get confused. So we're at 10 here, we're at 100 in the middle, and we're at 1,000 at the top. The red line there is 50. That's our magic number. We want breeds to be above it. And this is annual registrations, and again, it's logarithmic. So we've got 100 registrations, 1,000, 10,000, and so forth. All right, it's logarithmic. But if you look, just because you've got a lot of registrations, it doesn't necessarily mean you've got a big effective population size. You can have a lot of animals on the ground being registered per year, but still have a compromised effective population size. On the converse, you might not have many annual registrations, but you can have a perfectly healthy effective population size. If your population is small, let's suppose we've got an effective population size of 100. It will be losing diversity. It will lose through inbreeding, increasing inbreeding. We will lose 50% of diversity per generation. Sorry, 0.5% of diversity per generation. If we reduce the population by a half, we're losing 1%. That's part of the reason they chose 50, is because you lose 1% of your diversity per generation. And bear in mind, like I said, with horses, that's about every 9 years. With canines, that's every 2 or 3 years. If we halve it again down to 25, we've doubled it over here, so we're losing 2%. And by the time we get down to an effective population size of 6, we're losing 8% of our diversity per generation. Every 2 or 3 years with canines, you could be losing that much diversity. Just to look at that, I said we need to breed from as many males and females as possible. If we start to skew things, what happens? Let's take our effective population size, keep it at 100. If we've got an equal number of males and females, we've got an effective population size at 100, we know it's 0.5% effective population size. If we skew things, and in each of these cases we've still got 100 animals, if we get to a situation where we've got 25 stud dogs and 75 bitches we're breeding from, we've now got an enemy of 75 and we're losing 7%, yet we're at 0.7%, and carry that on into an equine situation where you might have 1 stallion and 99 mares, we've only got an effective population size of 4, and we're losing 12.5% of our diversity per generation. Quite important we breed from as many males and as many females as possible. So with our pedigree populations and all this background understanding, what do we do? Yeah, you can just divide the population up. I was talking to somebody earlier on, was talking about island populations. You can say, right, we'll take what we've got, we'll divide it into 3 groups, we'll send 1 group to America, stick a group in Japan, keep a group here, and there the twain shall meet. They will mutate, they will diverge, they will generate diversity between each of the groups. That works. It may be that one of those populations then becomes unviable because it's not mutating fast enough, and that's the point where you might have to dip into one of those other island populations, but you need to do it in an educated way. It's not just a case of taking any old stud dog and bringing it on its semen back across. Okay, and we can look at how we might manage it. Random mating, chilling and cattle, it works great. But the problem is with chilling and cattle, you know, calves are dying behind the hedge. And that kind of doesn't work in domestic livestock situation. You know, we have we get the vetting. Yeah? Management of inbreeding through a strategy called maximum avoidance of inbreeding. And that is pretty much the strategy that the Kennel Club is leading new hopes towards through their huge investment in computer software. If you go on the Kennel Club website for your breed, you can look at a stud dog, you can look at a picture, you can see what the inbreeding coefficient of the theoretical progeny is. That's great. It might start to control inbreeding, but certainly control it in your next generation. But there are inherent problems in that. The smaller the breed, the more of a problem it is, because it does nothing. Whilst it might reduce inbreeding, it does nothing to control loss of diversity. They're two different things. The way to do that, to control the loss of diversity, is to control inbreeding, which we've seen is linked to mean kinship, by managing mean kinship in the first place. Okay? And that is gold standard for management of breeds through pedigree. Management by mean kinship, control of rate of accumulation of mean kinship is the way to do it. That's gold standard. That's what happens in zoos around the world. It's increasingly what's happening in the rare equine world. What does it do? Manage mean kinship, you reduce the rate of inbreeding. It prevents crossing the rare blue Smarties with the common red Smarties, accepting it's a rare breed and everything is rare. But there are some genetics within that breed that will be rarer. And you need to try as much as you can to keep them apart. Because if you make an animal that's carrying a lot of red Smarties with one of those few animals that's got the blue Smarties, not only are you increasing the frequency of the rare genetics, the blue ones, you're also increasing the frequency of the red genetics, and it swamps it. And it actually puts the blue Smarties at even greater risk of being lost through genetic drift. So you need to make animals of very similar in kinship. That helps to maintain genetic diversity, and it also means that by using mean kinship, we can identify animals that are carrying the rarer genetics. Lower mean kinship, greater risk of being lost. If we were looking at a breed-wide program of cryopreservation in the equine world, which horses do we really need to line up first when there's limited funds for semen collection? And that's how it's been working in equines for a long time. With Cleveland Bays, there's a gene bank that's funded. It came out of foot and mouth. And yes, they took a snapshot across the breed to begin with for the doomsday scenario. But then going forwards, you make sure you've collected the animals of the lowest mean kinship, because they're the ones that are carrying the genetics at greatest risk of being lost. I've talked about the genetics, but I've told you that I'm a horse breeder myself, and I can look at something and I can go, it's got a leg at each corner, it tracks up really well, it's got 2 ears, it has to meet the breed standard, and it has to meet my knowledge and my understanding of the breed. Yeah? So there are 2 things in my breeder's toolbox. I've got phenotype, what's the horse like? You know, is it meeting breed standard? What's its stamp? Is it fit for purpose? And then we've got genotype. And that's all the bits that in the past we might have done by picking up the stud books and looking at pedigrees and trying to work things out and look for the outcross. But the problem is that with shuffling pieces of paper and stud books in those old days before home computers, you start going 5-generation pedigrees, yeah, that'll fit on an A4 sheet of paper. If it's really small print and I put the glasses on, I can possibly read an 8-generation pedigree. It's blooming small typeface. I've probably stuck 2 bits of A4 together which printed out A3. But we've got that Chinese grains of rice on the chessboard thing. It doubles. So in 10 generations, there are over 1,000 animals. You've probably on a windless day gone out onto your patio with a lot of sellotape to stick a lot of bits of paper together, or you're using a computer. Cleveland Bay stud book, at the time that my partner Jill and I started digitising the stud book, we traced it back 32 generations. Well, hang on a minute. If we go 20 generations, 10 generations, there's over 1,000 animals. At 20 generations, there's over 1 million animals. You're not going to Wembley Stadium to stick it all together. It just does not work. You need to start to use home computers and computer analysis to look at where those common chains of pedigree are, the common ancestors. And by the time you get down in Cleveland Bay to 32 generations, the number is outstanding. It's huge. It's over 4 billion for not million, billion animals. Well, wait a minute. The world horse population is only 58 million now. There's 4 billion there. They've got to be related. You know, they've got to be common ancestors. It's impossible. And the same will be true in the canine world as well. That if you step back beyond, you know, Adam and Eve were canines, where was it? It was before your stud book. Yeah, those animals will be related somewhere. So it's only through using computers to analyse the pedigree data that we can find this really, really deep inbreeding. Yeah, 5 generations, 32 animals, 8 generations, 256. I kind of think that's about where we stop sticking bits of paper together. 10 generations, we're out in the garden. 20 generations, you've got to be in computers. 32 generations, it's over 4 billion animals. In dogs' terms, best figure that I could find was about 470 million animals. So if you've got a 32 generation pedigree for your dogs, you've again exceeded the world dog population. They will be related. So how do you manage it? And a lot of people have heard of SPARKS, and there's some people in this room that actually used the very original version of SPARKS that was designed over in America, funded by Walt Disney. And the idea was that a zookeeper with a zookeeper's hat on, in charge of the whole world population of giant pandas, could work out what animals were related to each other, which were the least related to each other. We can move them around between zoos. We can mate animals of similar mean kinship, but that produce low inbreeding coefficients. That's great in its original form, as long as you've got one person in control. They can say, right, we're going to stick these animals on an airplane, we're going to ship them over to Munich Zoo, because that's where they are really compatible with the existing pandas that they've got, or whatever. What I did in the 1990s was took that original software and the science behind it, but turned it into something that we could actually provide to individual breeders. So it's not one person that's in charge, it's every Cleveland Bay mare owner responsible for their own breeding program. And certainly in the equine side of things, the right of the breeder is enshrined in law. It's enshrined in what was the EU Zootech regulations, and it's still enshrined in the UK animal breeding regulations. Breed societies cannot dictate to breeders what they do. They can provide advice, but they need to actually, put that advice out to breeders. So SPARKS, what we've done is we've identified different matings and whether they're advantageous, disadvantage, or actually damaging to the breed. Nowadays, using home computers, we've got it to a simple traffic light scheme. Green matings are really to be encouraged, because what they're doing is they're mating animals that have got similar mean kinship, but lower co-ancestry, lower inbreeding coefficient of the progeny than the mean kinship of the mare. There's a little twist in there, but that's basically lower than average inbreeding. Matings, co-ancestry, lower than a threshold we set for the breed as well. And every breed is different. With Cleveland Bays, we've had to pitch it at 24%. Not everybody can do that. It might be that there's no stallion anywhere near, so we've got to go for a best of the rest mating. And then still mating animals of similar mean kinship, but they're animals that are producing animals of higher co-ancestry than the mean kinship of the mare. But we're still mating animals that are of similar mean kinship to stop increasing the frequency of the red smarties as well as the blue smarties. They're preferred. If we can't do a green one, do a yellow one. They're not damaging for the breed, and we're getting foals on the ground. Now we get into the ones that are to be discouraged, and it's for exactly that blue-green smartie scenario, that we've got animals of significantly different mean kinship. We don't want people to do it, because actually, whilst it might be okay in this generation, it reduces options 1, 2, 3 generations down the road. We've got progeny of higher co-ancestry than both the parents. Co-ancestry is still lower than that breed threshold, 24% of Cleveland Bays. They bring together the common genetics and the rarer genetics, so they're not good matings. And then, of course, we've got red ones. These are the ones that actually produce inbreeding coefficients higher than the breed threshold. And we do not want them, because they're the ones that will actually increase the expression of those deleterious alleles in the breed. Different breeds, different thresholds. It's 24% for Cleveland Bays. Eriskay ponies, 18%. Suffolk's 12.5%. We've now got Shire horses on board. We've got Dell's ponies, Exmoor ponies. They've all got a different level. But, that's the red matings, we just really wanted to discourage them. And more importantly, what we want to do is to break chains of red matings. So if you've got a highly inbred colt that goes on to produce a highly inbred colt, that goes on to produce another highly inbred colt, that's when it's really going to go pear-shaped. So how's this work for Cleveland Bays? That's been in place with Cleveland Bays since 2004, much more recent with Eriskay's and Suffolk's, but it means we've run this experiment for over 16 years. That was the graph I showed you before, but I've just extended it into what's happened since the millennium. And if you look, that was the original graph. Now look what's happened. Inbreeding is still accumulating. It will do, it's a close stud book, but we'll reduce the rate of accumulation in breeding. And if you reduce the rate of accumulation in breeding, what happens? Is we increase effective population size. And we started this experiment. Look at where we were at the turn of the millennium. It was 19. If you remember, it's got to be 50 to survive. So it's bumped along. People have slowly taken it on board. At its worst, we had about 36% compliance with the green matings. At its best, and we're talking worldwide because it's a global stud book, 60%. So even if not 100% of people are doing green matings, we get an improvement. And look what's happened. By the time we get to around about 2014-15, it goes through the 50, and then it starts to increase logarithmically. Yeah? We've gone through the 100, and we're away. And provided we can control that rate of increase in inbreeding, we're controlling the loss of genetic diversity. Is that reflected in the number of foals we're getting on the ground year on year? No, it isn't. So it doesn't increase the number of foals that you're getting, but what it's doing is it's maintaining genetic diversity through the bad times, through the economic challenges of recent years. We've still got the diversity there to pass on to future generations of breeders to take the breeds forward. In terms of monitoring it, we need to use something like PopRep that I showed you before to make sure that it does continue to increase, that it doesn't go backwards. It will never be zero, yeah, unless we bring in a rate of increasing in breeding. It will never be zero unless we bring something in from outside and it will take a dip. But then are you breeding Cleveland Bay horses if you brought Clydesdales into the ring or whatever? Trying to reduce inbreeding really quickly, you'll get short-term gain and then it will rebound. It will go pear-shaped and it actually increases. All the models show you that. And that actually caps the maximum benefit. So if you only focus on maximum avoidance of inbreeding, which is what the Kennel Club is doing now, for the rarer breeds, that potential is compromising how much diversity you can preserve for the future. You need to manage it through linkage. So there we go for the future. What's pretty obvious is that uncoordinated breeding definitely leads to an unsustainable accumulation of inbreeding. We've shown that across species. That's got a really negative effect on effective populations. You need to understand your stud books. Get hold of the data and get them analysed. Do PopRep or something similar. Yeah. Then put an effective breed management plan in place. That might be SPARKS-like. There is a software company called Cloudlines who are taking the science of our version of SPARKS and putting it online. And that is increasingly going to become available for more and more equine breeds, but it will also work for canine breeds. It leaves the breeder in control, but the breed society still needs to have an umbrella view of what's going on. It avoids those highly damaging matings, maximises potential diversity, and it's another tool in the breeder's toolbox. Breed from as many males and females as possible. I've shown you what happens if you don't. If you skew it, you reduce effective population size. And avoid overusing a very small section of stud dogs in particular. If you've got a stud dog that breeds at cross, everybody wants it in its pedigree. Yeah, that isn't good for the breed as a whole. The science of these, we wrote up, Philippe Wilson and I and other colleagues at Nottingham Trent wrote up and submitted a paper. It's out there in Ecology and Evolution 2 years ago now. I think it's open access. It explains in a lot more detail than I have the science behind it and the results. If you want the link, it's there. You can quickly grab your phone and take a photograph of the link, or we can distribute the slides later. We got there. Any questions?